So in Chapter 8, we're going to look at DNA, not just the structure of DNA, but also the structure of chromosomes and some of the history of how DNA was determined to be the molecule of heredity. And so there will be some history that you're required to know, roughly a timeline of certain names, and then there will be a few experiments that you will need to know in detail. So as you go through this chapter, don't think that you're not required to know the details of the different experiments, because those will be covered on the test. And then for the other characters, know one sentence, for example, that describes what their important contribution was to the study of DNA. So a couple of important characters in the story. Before 1900, around late 1860s, we have Johann Miescher, and he was a doctor slash scientist who purified something from pus that he collected from bandages of people who had infections, and he extracted from the pus a material that he called nuclein, but it was an acidic material, and so what he really extracted was DNA. And so that was the first identification, I guess, of DNA chemically. Then Kossel, this was also before 1900. You have a scientist named Kossel who characterized that material that Miescher had described as nuclein. Kossel characterized it as containing 4 nitrogenous bases: adenine, cytosine, guanine, and thymine. So more chemical characterization of that molecule. Still, nobody knew that this was DNA, that this was the molecule of heredity. These are primarily biochemical descriptions of this material. And then, just after 1900, Phoebus Levine he was also looking at this material, and he proposed that DNA was a polymer of nucleotides, like Kossel had described, but that it consisted of a repeating tetranucleotide. So, like adenine, then guanine, then cytosine, then thymine, for example, and then repeat the same 4 again and again and again, all in a chain. And so you would end up with equal amounts of adenine, cytosine, guanine, and thymine, and that because of the repetitive nature of the tetranucleotide pattern along that DNA chain, it seemed at the time that DNA couldn't possibly be the molecule of heredity, because it didn't seem to have enough variability to provide what a molecule of heredity would need to provide, which is the ability to create all kinds of different traits and characteristics and so on. So Levine he was a good biochemist. Unfortunately, he's most famous for the thing he got wrong, which is called the tetranucleotide hypothesis. So there's some links here you can click on if you want to read a little more. Now, the first experiment that you're going to need to know and understand the experimental design, how it was set up and why, and then what the results were and the conclusions, is Fred Griffith's experiment in 1928. And his experiment dealt with a type of bacteria. It was a pneumonia-causing bacteria. He had 2 strains of it. The strains he called type 3S and type 2R. So I would pay most attention to the S and the R. The S stands for smooth, and the R stands for rough. And the difference between these 2 strains of this bacterium is that the smooth strain will grow into colonies on a plate that are nice and shiny and smooth. And the R strain will grow into colonies that are lumpy and not shiny and rough. And so it was a visual distinction between the 2. But also, what we know now is that the smooth strain has an extra capsule surrounding the cell that the rough strain doesn't have. So a capsule in a bacterium, a capsule is a coating of mostly carbohydrate on top of the cell wall, and it actually allows that bacterium to be better at evading a host's immune response. So it's a more dangerous type of bacterium if it has that capsule. So the smooth strain is called the virulent strain. It's a very aggressive and dangerous strain. And the R strain, because it doesn't have that extra capsule layer on the cell, the host can fight off an infection by the R strain. So the R strain of bacteria is non-virulent. And so what he did was he took some bacteria of the type S and he injected it into a mouse, and the mouse died. Okay, and then he did an autopsy and recovered live type S bacteria from the tissues of the mouse. Then he injected a mouse with type R bacteria, but the mouse was able to live. The mouse was able to kill off that infection. And they sacrificed that mouse and found that no bacteria are in the tissues of that mouse. Then they took type S bacteria, but then they boiled it. So heat killed type S bacteria. And they injected it into the mouse, and the mouse lives. And they sacrificed the mouse, and they find no bacteria in the tissues. The critical experiment, though, is shown here in letter D. You have a mixture of the heat-killed type S bacteria and the live type R bacteria mixed together. And by themselves, neither one of those were able to kill the mouse. But when you mix them together in the flask, and then you inject the mixture into the mouse dies, and they actually recovered live type S bacteria from the mouse tissues. Now you should recognize what's going on here, because we've already talked about transformation. Transformation of bacteria recombines genes from a dead cell, which enter then into a live cell and recombine with that chromosome and give you a new trait. So what's happening is the gene that encodes the capsule, or the enzyme that makes the capsule, has gone from a dead cell to a live cell and transformed that cell into a type S cell. So the type R has been transformed into type S by the transfer of a piece of DNA into that cell. And so that's what, but Griffith didn't know that's what he was seeing, but that's what is the explanation for this. So what he knows at this point is that something is going from one cell to another and causing the mouse to die of type S. But Griffith didn't really take this far enough in this experiment, so he never really could conclude that it was the DNA portion of the cell that was causing the transformation. So in 1944, another group, Avery-MacLeod-McCarty, published a paper similar, based on the Griffith's experiment, but they actually took it to the level where they could conclude that DNA is the molecule creating the transformation. So their question is, what's the chemical nature, meaning what type of molecule is what they called the transforming substance, the substance that could transform a type S cell, or excuse me, a type R cell into a type S. So what they did was they didn't use any live mice, but they used the same type of bacteria. They used type S bacteria. All right, they boiled it, then they broke the cells open, and they removed all the lipids and membrane pieces. And what you have left, once you remove all the membrane lipids and carbohydrates, what you have left in this mix, in this type S filtrate, would be a mixture of the different proteins, RNAs and DNAs that had been inside the type S cells. And the question is, which molecule then is it that is being transferred into the type R cells to create a transformation event? So what they did was they split this filtrate into 3 test tubes, and they used an enzyme called RNase in one test tube, and that will destroy all the RNA in that tube, leaving the protein and the DNA behind. In this test tube, they used protease in the center, and that will destroy all the proteins in that tube. And in this tube, they used DNase, which destroys DNA. So they had these enzymes, and they were able to target in each test tube one biomolecule for destruction. Then they mixed what was left in that test tube into a flask of type R bacteria. And what happened was the tube that had the RNA removed from it some of the cells still transformed into type S. So removing the RNA didn't stop the transformation event. The tube that had the protein removed from it, when mixed with type R cells, still caused some of the cells to transform into type S. So protein, when you remove the protein, that doesn't stop the transformation event. However, when you remove the DNA, this was the third tube where they used DNase to remove the DNA from the filtrate, and they mixed what was left into the type R cells. And then, I don't know how this cell got out of the bottle. I think that's a typo. But when they looked at that flask, none of the cells transformed into type S. So removing the DNA removes the ability of the cells to be transformed. So because only DNase destroyed the transforming substance, then it means that the transforming substance must be the DNA. So that's a classic experiment in 1944. And what's interesting about this is that it really, remember, you got to limit your conclusions. This means that in bacteria, DNA is the molecule of heredity, but you can't really extend that too far. However, subsequent experiments that were done in eukaryotes confirmed that DNA is also the molecule of heredity in eukaryotic cells. But at this time, they had just confirmed that DNA was the molecule controlling everything in bacteria. Now, in 1950, 6 years later, a scientist named Chargaff did a whole bunch of studies. Actually, there's many papers that he put out. But basically what he did was extracted DNA from every source he could get. This is 5 shown here, but there's several papers, and he has all these charts. So basically anything he could get his hands on, he extracted the DNA from those types of cells, and then he analyzed the composition of nucleotides in that DNA. So for example, when you extract DNA from E. coli and you break it down into the nucleotides, you see actually a pretty equal percentage of all 4 nucleotides from that DNA. But when you look at, for example, human DNA, you don't see equal amounts of all 4, but what you do see consistently is equal amounts of A and T, like sea urchin here, 32 and 32, and equal amounts of G's and C's. Okay? And so what he did so many of these, he didn't know what pattern he was looking for, but after he did a lot of this type of experiment, looking at all kinds of stuff, there's a lot of, basically he got whatever tissue he could get his hands on, which includes quite a bit of sperm from different animals. And but anyway, he was able to see in the data that there was this relationship between the amount of A and T, which was always about equal, and the amount of C and G was always about equal. And we know now that what he was really describing is the complementary base pairing rules, because every time you have an A on one strand of DNA, you have to have a T on the other strand in order for the double-stranded DNA. And so he was describing the base pairing rules. But he didn't know that at the time. He was just analyzing the makeup of DNA. The next big experiment came in 1952, a year before Watson and Crick would publish their structure of DNA. And this experiment is called the Hershey-Chase experiment, and it's based on, or it was using phage, bacteriophage, in their experiment. And so just briefly, I'm going to remind you that bacteriophage have a protein structure all the purple here is a protein, and then, or types of proteins, and then inside this head, remember the icosahedral head, is a chromosome of DNA usually, although there can be viruses that have RNA, for example. But the bacteriophage that they were using had a DNA chromosome inside a protein phage body. And here you can see a picture of many phages infecting the same cell. And you remember that the lytic life cycle involves the injection of the phage chromosome into the host cell, and a lot of times that phage chromosome will be cut up by the restriction enzymes that are already in the bacterium. But if it doesn't get cut up, then the phage will quickly reproduce itself and make an enzyme that will cut up the host chromosome. Then the phage genes will be expressed, proteins will be made, and new phage will be assembled, and the cell will lyse to release those phage. So that's the lytic life cycle. So what Hershey and Chase did, and what they published in 1952, is an experiment where they grew phage in a flask that had S35, that's an isotope of sulfur, and they also grew some phage in a flask that had P32, which is an isotope of phosphorus. And basically what they're doing is they're creating phage that have radioactive proteins, because protein contains sulfur, or phage that contain radioactive chromosomes, because DNA contains phosphorus. And it's really important that the DNA contains the radioactive phosphorus, but DNA doesn't have any sulfur in it. And then the protein contains the radioactive sulfur, but protein doesn't have phosphorus in it. So, in this case, so they were able to radioactively mark or tag the two types of biomolecules in the phage. Then they took those radioactive phage and in separate flasks, they infected host cells. And they let they left them in there long enough so that they could attach to the host cells and inject their whatever. They didn't know what was being injected into the cell. They didn't know if the protein was going into the cells to create the infection process or if the DNA was going in. So that's really the question. So they're letting the phage attach to the host cells, giving them enough time for the cells to become infected. And then they used a blender and a centrifuge to separate the cells from the phage. So the phage will be sort of torn off of the cells. And they studied what they got left. So when they centrifuge this, they get a solid portion at the bottom, which would be all the cells, and then a liquid portion at the top, which would be all the phage that had been shaken off. And what they noticed in those two sections was that for the phage that had been marked with S35, all the radioactivity was in the empty phage particles, and none of the radioactivity had entered the cells. Even though the cells were infected, they weren't radioactive. In the other tube, all of the empty phage were not radioactive, but all of the infected cells were. And so what it means is that the DNA had entered the cells to create the infection, not the protein part of the phage. And so this led to the conclusion that DNA is the genetic material in phage. And of course, we've gone on to show that DNA is the molecule that's controlling things in some viruses, although some viruses use RNA as well. And then, like I said, DNA was later shown to be the molecule that controls eukaryotic cell genetics as well. But the Avery-MacLeod-McCarty experiment and the Hershey-Chase experiment were both critical, and they both preceded the Watson and Crick publication of 1953, which describes the double helix structure of DNA. And lots of groups were working on this problem at the time, is what is the structure of DNA, because it was becoming increasingly obvious that DNA is the molecule of heredity, and so figuring out the biochemical structure of it in detail was important. And it was solved in 1953 by Watson and Crick. Watson and Crick didn't do any actual experiments, They actually talked to colleagues and people who were working on it and synthesized their results into a model that they built, literally with ring stands and clamps and molecule building kits, model kits. But they built their model with exact angles and so on. And they were able to come up with a model that matched all the data that they had. But none of the data that they had came from themselves. So one of the critical pieces of data that came was from a scientist named Rosalind Franklin, who was doing an X-ray crystallography of DNA. You have to get DNA to make a crystal. That's actually easy to do because DNA forms a crystal pretty nicely. Then you have to shine X-rays at the crystal, and the X-rays diffract off of the atoms in the molecule, and they hit an X-ray screen or an X-ray film, and they make little dots and spots, and then by measurements and calculations from that film you can figure out the position of every atom in the molecule. People still do this, and I mean the technology hasn't really improved very much, more or less, but anyway, people still do this to figure out the structure of three-dimensional structure of molecules. So this is a picture of Rosalind Franklin, and she worked with a guy in her lab named Maurice Wilkins, but they didn't get along. And the story goes that when Rosalind Franklin wasn't there, Watson and Crick came by and talked to Maurice Wilkins, who told them that there was a pretty good piece of data that Franklin had come up with, and he showed them the data. Wilkins showed Watson and Crick the X-ray picture, and just by looking at that X-ray picture they realized that it had to be a double helix with two strands that were equally spaced all the way down the helix, which was a question they had still been wondering about. So in any case, in 1962 the Nobel Prize was awarded to Watson and Crick and Wilkins, despite the fact that even maybe unknowingly or unwillingly, Rosalind Franklin had provided the X-ray data that really put things into place. And the reason she didn't win the Nobel Prize was not because she's a woman, but because she had died early. She had died young. You can see in this picture she's young. And she had died from cancer, probably because of too much exposure to X-ray, possibly. But she had died of cancer, and they don't award the Nobel Prize posthumously. So one thing students always think is that the Nobel Prize is the same thing as the Nobel Peace Prize, and that's not the same thing. The Nobel Peace Prize, it's given by the same group, but the Nobel Prize is, the full name of it is the Nobel Prize in Medicine or Physiology, and then there's also a Nobel Prize in Chemistry. And so those are not the Peace Prize. Those are our academic prizes for big discoveries. The Nobel Peace Prize usually goes to someone like Mother Teresa or, for some reason, Barack Obama. But the scientific prize is just called the Nobel Prize in Medicine or the Nobel Prize in Chemistry. So there's two different ones that we will typically talk about in this class. All right, so what is the structure of DNA? I think I will end this section and pick this up on the next lecture section.